Assalamualaikum and hi. Okay, today I'm Siti Nur Aliya binti Johari. My number matrix is D2018208593. We'll explain about experiment 2 about photosynthesis, and the title of this experiment is rate of photosynthesis. The objective of this experiment is to determine the effects of light intensity on the rate of photosynthesis. For the introduction, photosynthesis, the process by which green plant and certain other organisms transform light energy into chemical energy. Plant has chloroplasts that contain chlorophyll pigment, which is this pigment react to light. The Calvin cycle has 4 main steps. First, carbon fixations. 2, reduction phase. 3, carbohydrate formations. 4, regeneration phase. Okay, so the energy to full chemical reactions in the sugar generation process is provided by ATP and NADPH, chemical compound which contain the energy plants have captured for the light. 3 main factors can limit the rate of photosynthesis. First is light intensity. Second is carbon dioxide concentrations. 3 is temperature. Hypothesis: the light intensity increase, the rate of photosynthesis will increase too.
In this experiment, we use plant sodium bicarbonate is the water container, hole punch, table lamp, ruler, and lastly aluminium foil. The plant is used in this experiment is bougainvillea, which is known as pokok bunga kertas. After we take the plant and we make hole with hole punch, so we get only 10 leaf discs from the plant. We take the syringe and we pull out the plug from the syringe and place the 10 leaf discs in the syringe. And then we replace back the plug into the syringe and carefully do not damage the leaf. And then we have water, plain water, and we put the sodium bicarbonate in the water and it becomes solution of sodium bicarbonate. And then we draw about 6 until 8 ml of solution bicarbonate solutions into the syringe. And then we remove the bubble in the syringe. We hold the syringe with a tip up and expel the water gently by pushing the plug until there no air in the syringe. We pull and as a vacuum for a few seconds and release the plug, letting it snap back and until there no air in the syringe. After there no air in the syringe, so we put back into the solutions of sodium bicarbonate and we measure with rulers 3 cm light above the container. So we observe it, and after 40 minutes we see the leaves. This is floating. So this is the result, and then we continue with 10 cm light. So we see only seed leaf floating after 40 minutes when the 10 cm light above it. So we continue in the dark place and cover with aluminium foil so we can see our leaf sinks in the container.
The graph, we can see the difference. The upper is from 3 cm light from the container, and the down is 10 cm light from the container. So we can see at 3 cm, the graph is linear, which all the leaf is floating, while at the 10 cm we have four leaf do not floating but sink in the ground after 40-minute observations. Discussion: how does the suctions help the leaf to sink? Okay, when the suction we act like a vacuum to the leaf this in the solution, the air in the leaf is forced out and replaced with solution that causes the leaf to sink. Okay, the second is, if you conduct the experiment without baking soda, what will happen to the leaf? This why? Okay, the leaf not floating because the baking soda is provide this of carbon dioxide, and when do not have a baking soda, so the leaf cannot floating. Okay, the third is why do the leaf this is in the baking soda solution treatment begin to float. So the plant material will generally floating in the water. This is because leaf have air in the space between the cell, which it had them collect the carbon dioxide gas from the solutions. Okay, what will happen to the leaf this in the dark? Justify your answer. When place floating leaf this in the dark, okay, we have a floating leaf after we have doing the experiment, which is a light between 3 cm and 10 cm from the container, the leaf will floating, right? So when we place in the dark, the leaves will sink because without light we have no energy, photosynthesis will occur, so no more oxygen gas will be produced in when we put leaf in the dark place. As conclusions, we can see the hypothesis is accepted, which is the light intensity increase and the rate of photosynthesis also decrease because the light is source of energy for the plant. So when have light, the rate photosynthesis will increase too.